नमस्कार मित्रांनो, माय बेस्ट गुरु चॅनल मध्ये आपले मनापासून स्वागत. आम्हाला प्रोत्साहन देण्यासाठी सबस्क्राइब बटनवर क्लिक करून सबस्क्राइब करून आम्हाला मदत करा, म्हणजे आम्ही तुमच्यासाठी घेऊन येऊ जास्तीत जास्त व्हिडिओज. तसेच आमच्या नवीन व्हिडिओचे नोटिफिकेशन मिळण्यासाठी घंटीच्या चित्रावर क्लिक करा. Dear students, lesson number 16, Heredity and Variation. Important topics in this lesson: inheritance, heredity, characteristics and their appearance, Mendel's laws of inheritance, diseases due to chromosomal aberrations. Question number 1. Complete the following sentences by choosing the appropriate words from the bracket. Inheritance, sexual reproduction, asexual reproduction, chromosomes, DNA, RNA, gene. A. Hereditary characters are transferred from parents to offsprings by dash. Hence they are said to be structural and functional units of heredity. Genes. Hereditary characters are transferred from parents to offspring by genes. Hence they are said to be structural and functional units of heredity. B. Organisms produced by dash show minor variations. Asexual reproduction. Organisms produced by asexual reproduction show minor variations. C. The component which is in the nuclei of cells and carries the hereditary characteristics is called dash. Chromosomes. The component which is in the nuclei of cells and carries the hereditary characteristics is called chromosomes. D. Chromosomes are mainly made up of dash. DNA. Chromosomes are mainly made up of DNA. E. Organisms produced through dash show major variations. Sexual reproduction. Organisms produced through sexual reproduction show major variations. Question number 2. Explain the following. A. Explain Mendel's monohybrid progeny with the help of any one cross. Answer. In this experiment, Mendel brought about the cross between two pea plants with only one pair of contrasting characters. This type of cross is called a monohybrid cross. So as to study the monohybrid ratio, let us consider the characteristic plant height with a pair of contrasting characteristics: tall plant and dwarf plant. Parental generation P1. Tall pea plants and dwarf pea plants were used in this cross. Hence, this is parent generation P1. Mendel referred to the tall and dwarf plants as dominant and recessive respectively. Though all the plants in F1 generation were tall, they contained the factor responsible for dwarfness. Though the phenotype of F1 plant is tall, their genotype is mixed. In the second filial generation F2, both as tall and dwarf types of plants appeared. According to the data collected by Mendel, out of 929 pea plants, 705 were tall and 224 were short. Thus the phenotypic ratio of these plants is 3 tall, 1 dwarf, and genotypic ratio is 1 tt is to 2 t small t and 1 small tt. Capital T shows dominant and small t shows recessive. Let us see Mendel's experiment of the monohybrid cross. In parental generation P1, phenotype is tall versus dwarf. Genotype in tall both are capital letters T and dwarf small letters TT. Gametes are, one is given from tall dominant, that is capital T, and one is given small t by the recessive dwarf. So first filial generation F1, 1 capital T, 1 small t. Phenotype. So the phenotype in this is tall. Now parental generation P2, selfic in F1, phenotype is tall and tall. Genotype is capital T and small t, capital T and small t. Now gametes are either capital T and or small t, in second capital T or small t. So generation F2. So female gametes capital T small t and male gametes capital T small t. Combining them, first capital T phenotype tall, capital T small t phenotype tall, capital T small t phenotype tall, and small t dwarf. B. Explain Mendel's dihybrid ratio with the help of any one cross. In the dihybrid cross, two pairs of contrasting characteristics are under consideration. Mendel performed more experiments on hybridization, in which he considered more than one pair of contrasting characteristics. He brought about a cross between a pea plant producing rounded and yellow colored seeds and pea plant with wrinkled and green colored seeds. In this cross, two pairs of contrasting characteristics were considered: color of seeds and shape of seeds. Hence it is called dihybrid cross. Parental generation P1. Mendel selected the pea plants producing rounded yellow seeds and wrinkled green seeds as parent plants. During gamete formation in P1 generation, the pair of gametes separate independently as in capital R, capital Y plants. Only capital R, capital Y type gametes are formed and not capital R and capital Y. Similarly, in small r, small y plants, only small r, small y gametes are formed. Mendel expected that in the F2 generation of dihybrid cross, plants would produce rounded yellow seeds. He was proved right. Though the genotype of these plants was capital R, capital Y, small y, their phenotype was like the parents, producing rounded yellow seeds. Plants of the F1 generation of dihybrid cross produce four types of gametes: capital R, small y, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Of these gametes, capital R, capital Y and small r, small y are similar to those of the P1 generation. F2 generation is formed through the selfing of F1 plants. Mendel's experiment of dihybrid cross. Parental generation P1. Phenotype rounded yellow seeds, wrinkled green seeds. While genotype capital R, capital Y, small r, y. Gametes from these are capital R, capital Y, and from wrinkled green seeds, small r, y. Now in first filial generation F1, capital R, small r, and capital Y, small y. Now parental phenotype rounded yellow seeds. Parental generation P2, selfing in F1, phenotype is rounded yellow seeds and rounded yellow seeds. Genotype capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. Gametes are capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So in second filial generation F2, kindly see the table for details. C. Distinguish between monohybrid and dihybrid cross. Monohybrid, dihybrid. 1. The cross involving only one pair of allele is called as monohybrid cross. The cross involving two pairs of allele is called as dihybrid cross. 2. The phenotypic ratio of monohybrid cross is 3:1. The phenotypic ratio of dihybrid cross is 9:3:3:1. 3. A monohybrid cross is useful in determining the dominance of genes. A dihybrid cross is useful in studying the assortment of the offspring. D. Is it right to avoid living with a person suffering from a genetic disorder? No, it is not right to avoid living with. No, it is not right to avoid living with a person suffering from a genetic disorder, because genetic disorders are non-communicable diseases. It means they cannot transfer from one person to the other. Question number 3. Answer the following questions in your own words. A. What is meant by chromosome? Explain its types. Answer. The structure in the nucleus of cells that carries the hereditary characteristics is called the chromosome. It is made up mainly of nucleic acids and proteins. There are 4 types of chromosomes. Types of chromosomes. 1. Metacentric. The centromere is exactly at the midpoint in this chromosome, and therefore the chromosome looks like the English letter V. The arms of this chromosome are equal in length. 2. Submetacentric. The centromere is somewhere near the midpoint in this chromosome, which therefore looks like English letter L. One arm is slightly shorter than the other. 3. Acrocentric. The centromere is near one end of this chromosome, which therefore looks like the English letter J. One arm is much smaller than other. 4. Telocentric. The centromere is right at the end of this chromosome, making the chromosome look like the English letter R. This chromosome consists of only one arm. B. Describe the structure of DNA molecule. Answer. The structure of the DNA molecule is the same in all organisms. In 1953, Watson and Crick produced a model of the DNA molecule. As per this model, two parallel threads of nucleotides are coiled around each other. This arrangement is called a double helix. This structure can be compared with a coiled and flexible ladder. Each strand in the molecule of DNA is made up of many small molecules known as nucleotide. There are 4 types of nitrogenous bases: adenine, guanine, cytosine and thymine. Adenine and guanine are called as purines, while cytosine and thymine are called pyrimidines. In the structure of the nucleotide, a molecule of nitrogenous base and phosphoric acid are each joined to a molecule of sugar. As there are 4 types of nitrogenous bases, nucleotides also are of 4 types. Nucleotides are arranged like a chain in a molecule of DNA. The 2 threads of DNA molecule are comparable to the 2 rails of a ladder, and each rail is made up of alternately joined molecules of sugar and phosphoric acid. Each rung of the ladder is a pair of nitrogenous bases joined by hydrogen bonds. Adenine always pairs with thymine and cytosine always pairs with guanine. C. Express your opinion about the use of DNA fingerprinting. Answer. The sequence of the genes in DNA of person as the genome of the person is identified. It is useful to identify the lineage and to identify criminals because it is unique to every person. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. A DNA sample taken from a crime scene is compared with a DNA sample from a suspect. If the 2 DNA profiles are a match, then the evidence came from that suspect. D. Explain the structure, function and types of RNA. Answer. RNA is the second important nucleic acid of the cell. This nucleic acid is made up of ribose sugar, phosphate molecules and 4 types of nitrogenous bases: adenine, guanine, cytosine and uracil. The nucleotide as smallest unit of the chain of the RNA molecule is formed by the combination of ribose sugar, phosphate molecule and one of the nitrogenous bases. Large number of nucleotides are bonded together to form the macromolecule of RNA. According to function, there are 3 types of RNA. 1. Ribosomal RNA. The molecule of RNA which is a component of the ribosome organelle is called ribosomal RNA. Ribosomes perform the function of the protein synthesis. 2. Messenger RNA. The RNA molecule that carries the information of protein synthesis from genes, as in DNA chain in the cell nucleus to ribosomes in the cytoplasm which produce the proteins is called messenger RNA. 3. Transfer RNA, tRNA. The RNA molecule which, according to the messenger of the RNA, Messenger mRNA carries the amino acid up to the ribosomes is called transfer RNA. E. Why is it necessary for people to have their blood examined before marriage? Answer. It is necessary for people to have their blood examined before marriage because by performing blood tests on prospective couple, the chances of having abnormal children can be understood. The genetic disorders cannot be cured any time. It comes with the birth and ends with the death. Therefore, it is necessary for people to have their blood examined before the marriage. Question number 4. Write a brief note on age. A. Down syndrome. Answer. 1. Down syndrome is a disorder arising due to chromosomal abnormality. This is the first discovered and described chromosomal disorder in human beings. This disorder is characterized by the presence of 47 chromosomes. 2. It is described as trisomy of the 21st chromosome. Infants with this disorder have extra chromosome with the 21st pair in every cell of their body. Therefore, they have 47 chromosomes instead of 46. 3. Children suffering from Down syndrome are usually mentally retarded and have a short lifespan. Mental retardation is the most prominent characteristic. Other symptoms include short height, short wide neck, flat nose, short fingers, scanty hair, single horizontal crease on palm, and a life expectancy of about 16 to 20 years. B. Monogenic disorders. Answer. Disorders are diseases occurring due to mutation in any single gene into a defective one are called monogenic disorders. Approximately 4,000 different disorders of this type are known. Due to abnormal genes, their products are either produced in insufficient quantity or not at all. It causes abnormal metabolism that may lead to death at a tender age. Examples of such disorders are Hutchinson's disease, Tay-Sachs disease, galactosemia, phenylketonuria, sickle cell anemia, cystic fibrosis, albinism, hemophilia, night blindness, etc. C. Sickle cell anemia. Symptoms and treatment. Answer. Normal hemoglobin has glutamic acid as the in this sickle cell anemia is C. Sickle cell anemia. Symptoms and treatment. Answer. Normal hemoglobin has glutamic acid as the sixth amino acid in its molecular structure. However, if it is replaced by valine and the shape or structure of the hemoglobin molecule changes, due to this, the erythrocytes or red blood corpuscles (RBC), which are normally biconcave, become sickle shaped. This condition is called sickle cell anemia. The oxygen carrying capacity of hemoglobin in such individuals is very low. In this condition, clumping and thereby destruction of erythrocytes occurs most often. As a result, blood vessels are obstructed and circulatory system, brain, lungs, kidneys, etc. are damaged. Sickle cell anemia is a hereditary disease. It occurs due to changes in genes during conception. If the father and mother are both affected by sickle cell anemia, or if they are carriers of this disorder, their offsprings are likely to suffer from this disease. Hence, marriages between persons who are carriers of or suffering from sickle cell anemia should be avoided. There are two types of persons affected by sickle cell anemia. 1. Sickle cell anemia carrier AS. 2. Sufferer from sickle cell anemia SS. Symptoms of sickle cell anemia: swelling of hands and legs, pain in joints, severe general body aches, frequent colds and cough, constant low-grade fever, exhaustion, pale face, low hemoglobin content. Treatment or remedies for sickle cell anemia. 1. Husband and wife should get their blood examined before marriage or after it. 2. A carrier or sufferer should avoid marriage with another carrier or sufferer. 3. A person suffering from sickle cell anemia should take a tablet of folic acid daily. Question number 5. How are the items group A, B and C interrelated? Answer. 1. Leber hereditary optic neuropathy, mitochondrial disorder. This disorder arises during development of zygote. 2. Diabetes, polygenic disorder, effect on blood glucose level. 3. Albinism, monogenic disorder, pale skin, white hair. 4. Turner syndrome, 45 plus X, women are sterile. 5. Klinefelter syndrome, 44 plus XXY, men are sterile. Question number 6. Fill in the blanks based on the given relationship. A. 44 plus X is to Turner syndrome, so as 44 plus XXY is to Klinefelter's syndrome. 3:1 is to monohybrid, so as 9:3 is to 3:1 is to dihybrid. C. Women is to Turner syndrome, while men is to Klinefelter's syndrome. Question number 7. Complete the tree diagram below based on the type of hereditary disorders. Answer. Hereditary disorders: chromosomal abnormalities, monogenic disorder, mitochondrial disorder, polygenic disorder. Now in chromosomal abnormalities: 1. Down syndrome, 2. Turner syndrome, 3. Klinefelter syndrome. In monogenic disorder: Hutchinson's disease, Tay-Sachs disease, galactosemia, sickle cell anemia, albinism, hemophilia, night blindness, etc. In mitochondrial disorder, Leber hereditary optic neuropathy. In polygenic disorder, cleft lip, cleft palate, constricted stomach, spina bifida, diabetes, blood pressure, heart disorder, asthma, obesity.
आम्हाला प्रोत्साहन देण्यासाठी सबस्क्राइब बटनवर क्लिक करून सबस्क्राइब करून आम्हाला मदत करा, म्हणजे आम्ही तुमच्यासाठी घेऊन येऊ जास्तीत जास्त व्हिडिओज. तसेच आमच्या नवीन व्हिडिओचे नोटिफिकेशन मिळण्यासाठी घंटीच्या चित्रावर क्लिक करा.